Welcome, everybody. It is fantastic to see you all here. My name is Alika. Welcome, welcome. It truly is an honor that I get to officially welcome you to the 2024 IMGC Roll Up Your Sleeves workshop. We have an incredible lineup of speakers for you today. A wealth of knowledge from quite literally around the entire world. I am going to kick us off with a rapid introduction, and then I am going to pass it off to our moderator, who is going to introduce our first speaker. How our session is going to work today is we have 4 speakers. Each speaker will present 25 minutes, a brilliant presentation about the process of scaling up, analyzing, and navigating the regulatory aspects, all about bioactive milk lipids. After all 4 of the speakers have given their presentation, we will have a 5-minute break. And at that point, we'll come back and have a 1-hour Q&A discussion where we'll bring all the panelists back onto the screen, and Dr. Bruce German will be moderating. Before we get there, I am Alika. I am this year's workshop chair. It's been a joy working very closely with this group of brilliant experts for the last 6 months, bringing this event to life. My journey with IMGC has been, it started in 2014 when I was a graduate student myself, and I presented my dairy-based dissertation research at the annual symposium in Aarhus. Since then, my relationship with IMGC has continued to grow very steadfast. I've worn multiple hats, from being a graduate member to the Splash content strategist, and now serving as the chair for this workshop. So I'm really thrilled to virtually be here with all of you and together learn about bioactive milk lipids in today's workshop. On this next slide, I want to quickly introduce you to who IMGC is. If you're new to IMGC, welcome. IMGC stands for the International Milk Genomics Consortium. It's a scientific society that was created in 2004. It was created by Dr. Bruce German, who we are very lucky to actually have as our moderator here today. And the original goal of IMGC was to sequence the lactation genome. Now, this was completed in 2009. You see the scientific references for this at the bottom of the slide. And when that was completed, this group of scientists realized our work was not done. There's still so much to learn about dairy. So we're going to expand the goal, and today the goal of IMGC is to advance the field of lactation and milk science to improve human health. There's hundreds of scientists who are part of IMGC. We hope that if you are not a member, that you will be after today's workshop. Quickly, you also notice that there are 2 logos on the screen. You see UC Davis and the California Dairy Research Federation. That's because this organization is co-managed by these 2 organizations. Now, IMGC consists of 4 main pillars, which is how, through these 4 pillars, is how we advance the field of milk science. The primary pillar, which is the first pictures that you see here, this is our annual conference, or the annual symposium. Here we have scientific discoveries presented by research every single year. Every other year, this conference is hosted at UC Davis. The years in between, it is hosted at a global location. So this year in October, IMGC Symposium will be hosted at UC Davis. Next, we have an awards program. Through this program, we offer awards to students as well as legacy career awards. These awards are handed out at the symposium. Then we have a translational arm, which is called the Splash Milk Science newsletter. This is a bi-monthly newsletter where new research is translated, written and translated by expert scientific writers. It can hit your inbox so you can stay up to date on all the latest discoveries in the milk science world. Highly recommend you subscribe if you're not a subscriber yet. And then last but not least, this webinar that we are all in today, the annual Roll Up One Sleeves workshop. So you will walk away from today's session with practical how-tos that you can apply to either your work or share with your colleagues, and hopefully it help advance the field of dairy science. So for today, we have a lineup of incredible experts. We have Dr. Bruce German as our moderator. I mentioned that he is the one who founded IMGC back in 2004. He is going to pop on the screen in just a little bit and we'll introduce our first speaker. I won't read all the bios of our four speakers because that would take up almost the entire time. They are long, they are brilliant, but I do want to really emphasize that this is a global initiative. We have not only all of you calling in from all over the world, our speakers are calling in from all over the world too. We're going to kick things off with Marie-Caroline in just a few minutes. She is calling in from France. Then our second speaker is Nurit. She's calling in from Israel. Our third speaker will be Ben. He's calling in from Sweden, going to Australia in just a few days. And then we have Amir, who is calling in from UC Davis. You also see a QR code on this screen. And I know that typically we ask you to put your phones away, but please grab your phone, scan this QR code so that you have it at the ready. We really want to hear your feedback. So either after every presenter is done, take that survey about that presenter, or at the end of the entire workshop, it will take 5 minutes to complete the entire session. Without further ado, let's welcome Dr. Bruce German so he can say a quick hello, and then we'll pass it over to Marie-Caroline.
Good morning, good afternoon, good evening. I'm delighted to be here. If I was to say who would I like to spend a day with talking about milk lipids, the four people in the world that I would most like to talk with are here today. This is the lottery for lipid geeks. And if you're like me, you paid no attention to that thing going on yesterday with the moon and the sun. You were getting some sleep in preparation for today. It's my great pleasure to introduce our first speaker, Marie-Caroline Michalski. Please, Marie-Caroline, take it away.
So I'm really delighted to talk today and introduce this great event by maybe insights of a past journey of mine from milk fat globule structure and physicochemical properties in foods to the dairy matrix effect in nutrition and health. These are my disclosures. So the quick background for this is that I am now very much interested in the nutritional and health impact of milk lipids, and especially of their complex structures in dairy foods. And this topic and the very much exciting new insights are based on the fact that nutritionally, for a long time ago, milk fat has long been considered merely as a rich source of saturated fatty acids. But in fact, in the recent years, we have discovered that milk fat is much more than that, even in nutrition and health. And interest about the milk fat globules and their unique properties has emerged from their initial importance in the dairy functional properties, but now also to their importance in the nutritional and metabolic effects of dairy products. And this led to the dairy matrix effect concept in nutrition and health. So I will take you through this journey through a few examples of questions and solutions that were found to explore the properties and impact of these milk fat globule structures beyond their simple fatty acid composition. In the beginning of my career, when I was hired at the Inrae Institute, my first challenge to tackle was to understand how the dairy processes could impact not only the structure of the milk fat globule, but also their functional properties. And it was known by that time that the structure of native milk fat globules, large size, a few micrometers large, covered by a biological milk fat globule membrane, was very different from the structures generated by homogenization process, submicronic particles with a membrane that get covers with caseins and whey proteins and inducing therefore a milk fat globule membrane damage. It was known that the damage of the milk fat globule membrane increased in dairy processing problems of cramping, coalescence, lipolysis, oxidation, fouling of dairy processing membranes, etc. And that it could modify the functional properties of dairy. However, there was a question, a technical point to solve. It was how can we really analyze the extent to which the milk fat globule membrane can be damaged, and how to assess the relative impact of milk fat globule size versus the interface composition that would be more or less damaged. So the first solution that I envisioned by that time was to use some described properties of different electrokinetic potential of the milk fat globules that were known by them to be minus 13 millivolts for the milk fat globule membrane and much lower, minus 20 millivolts for casein micelles. So I thought that maybe using the laser Doppler electrophoresis technique that is used to measure this electrokinetic surface potential of the droplets, maybe there could be a gradual increase of this zeta potential, electrokinetic potential, representing kind of the surface charge of the particle, when the interfacial coverage with caseins would increase. So I coupled measurement of particle size analysis using laser light scattering with zeta potential, electrokinetic potential surface measurement according to gradual increase of the damage by increasing homogenization pressure. And indeed, we could observe that as much as the fat globule size and damage decreased, then the apparent zeta potential with casein coverage increased. And of course, it is known that when the particle size decreased, this increased the specific surface area of the milk fat globules, because when globules are more small, there is more surface relative to the interior. So this led us to be able to calculate, to a model to calculate the proportion of the surface of the milk fat globule that is covered by proteins, caseins, and mostly, rather than by the milk fat globule membrane. And so this allowed to generate a calculation model to merely by measuring the zeta potential of the milk fat globule to be able to estimate the percent of damage, which means the percent of surface that is covered with caseins. And then we also, in order to decipher the relative impact of the size and the damage on the properties of the dairy product, we optimize a microfiltration process on a membrane in order to separate native milk fat globules according to their size. And so we could thereby obtain the largest milk fat globule fraction, 6 microns about approximately, and also the smallest ones below 1 micrometer in diameter. But the difference with homogenized ones is that small fat globules collected by microfiltration of whole milk are in fact in their native state covered with the milk fat globule membrane, which we could check by measuring their zeta potential that was still intact at minus 13 millivolts. And this could lead to the development of many follow-up advances on the importance of milk fat globule size on their biochemical, physicochemical properties. And finally, we set up an experimental design in order to really decipher how the relative impact of the size of the milk fat globules and of their surface damage. So there was here the utilization of either cross-flow microfiltration here in order to have small to large milk fat globules that are native. Then homogenization could be used at different intensities to have different combination of size and surface damage. And also we could simply emulsify anhydrous milk fat in proteins in order to have different size of globules that are all entirely damaged. And then we prepare drainette and acid gels with these milk fat globules of different sizes and surface damage. And we analyzed their physicochemical properties by rheometry and the structure of the milk fat within by confocal microscopy. And what we could reveal in this a combination of approaches is that in fact there is a threshold of the alteration of the milk fat globule surface, beyond which the elastic properties of the gel increases, which means that when the milk fat globules are sufficiently small and when their membrane is native, they can act as an inert filler within the protein matrix. However, when they are native but they are too big, they can act as a structure breaker and the gel gets softer. This is the image here. And when they are very small but very damaged, in fact, they contribute to the protein network and they act as a structure promoter and enhance the viscoelastic properties of the dairy gel. So it was really the combination of these different physicochemistry, zeta potential, microfiltration approaches, and rheology that allowed to decipher the impact of this milk fat globule structure on the physicochemical properties, rheological properties of the product. But then after all this work about really the functional properties of the dairy product, let's say, the question was, but would this also maybe impact the fate of the dairy fat during the digestion, intestinal absorption, and would, for example, homogenized milk fat globule be induce different absorption and metabolization of the milk fatty acids? This was an important question by that time, and this raised the question of how to track the ingested fatty acids from milk fat in preclinical studies in humans. So the first solution that we elaborated was thanks to a chat with my animal physiology colleagues. So it's always very useful to have a interdisciplinary discussion with other scientists. We found the solution of incorporating a 13C triglyceride tracer in anhydrous milk fat, which then, after elaborating large droplets covered by phospholipids to mimic native milk fat globules, or small droplets covered by caseins to mimic small homogenized milk fat globule, and by feeding this as an intragastric gavage to rats, then you can monitor the appearance of the 13C tracer in the expired CO2 of the animal. And this provides you a global insight of how the ingested structure of the dietary fat can potentially modify the final beta oxidation of the ingested fatty acids. And this is what we could observe in this experiment, in which finally there was a faster appearance of 13C in the breast of animals when the same anhydrous milk fat labeled with 13C was actually administered in large droplets covered by phospholipids mimicking the native milk fat globules. So after this very encouraging proof of concept, our challenge was how could we prove this in humans? So this was a collaboration with the Human Nutrition Research Center, specialized in performing postprandial tests.
And so we could develop approximately the same kind of approach, but by testing in humans the two most extreme structures, which was unemulsified anhydrous milk fat compared to very finely homogenized anhydrous milk fat in skim milk. We wanted to track very precisely the fate of this fat after digestion and intestinal absorption, fecal loss, and final beta oxidation in lean and also in overweight and obese volunteers. So our strategy to really conclude about the supramolecular structure was to have two real meals with exactly the same composition. There was the same amount of milk fat, the same amount of skim milk, and the same amount of bread. However, the milk fat was either dispersed in the skim milk or spread on bread. That was the only difference. And there again, in order to track the fate of the fatty acids, in fact, we enriched milk fat with 13C triglycerides with different type of molecule that would represent exactly the proportion of the medium, long chain saturated and unsaturated fatty acids in milk fat. So tryptanoin, tripalmitin, and triolein labeled with 13C.
So this way, when our volunteers came each time at the Human Nutrition Research Center to have these different test breakfasts, including the 13C tracers, we could collect plasma, postprandial chylomicrons, but also performed breath tests with indirect calorimetry by measuring the gaseous exchange. And this way, it allows to track the fate of the tracers in the breast, just as we did in our preclinical trial. But one question was, okay, maybe we could
measure different beta oxidation in breast, but there could be a bias if in fact our tracer was simply more lost in stools. So this is why we decided also to collect the stools of the volunteers over 3 days after ingestion of the test breakfast. And we could, in fact, estimate that there was some elimination of the tracers in the feces, which allowed to refine our calculation. And in this way, we could really precisely measure not only that the emulsified structure of the fat can stimulate a rapid intestinal absorption and even a better return to baseline in obese persons,
but also that the structure of the chylomicrons was different between lean and overweight subjects, which can also contribute to this better metabolization here, the return to baseline of the postprandial lipemia in the obese volunteers. And finally, using the tracers, we could observe that when the same fat was emulsified instead of bulk, this increased the release in plasma of labeled free fatty acids that were finally more beta oxidized by the body. So using the breast test. So using this combination of formulation approach and enrichment with tracer, we could show that emulsification finally modulates the final use of the ingested fatty acids by the body. So it is really an interesting perspective that according to the fat structure in the foods, and this is consistent for dairy foods, there can be a faster or higher use of the dietary fatty acids as an energy source during the hours after the meal when they are brought as an emulsified form.
And our last challenge was related to better characterizing the potential nutritional interest of the milk fat globule membrane, because of course milk fat globules are emulsified structures, but they are unique structures surrounded by this milk fat globule membrane. And there were 10 years ago several preclinical data on animal models showing that the milk fat globule membrane may reduce cholesterolemia. But by that time, there were only few clinical trials that were interesting and seemed to have a neutral or moderate effect, but mostly in healthy volunteers and not necessarily very significant. We were wondering what would be the impact of milk PL on postprandial metabolism in real dairy products in subjects at cardiovascular disease risk. And to study this topic, we also wanted to envision a sustainability content. And by that time, there was a beginning to be interest about buttermilk that could be valued as a rich source of milk PL, but the real metabolic benefits remain to be demonstrated. And also, because preclinical data were mainly about cholesterol, there were questions about the characterization, fate, and role of milk sphingolipids, and also impact on other metabolic endpoints of interest, like the gut microbiota or inflammation, for example. So this was quite a huge topic to tackle to begin with. And in order to really develop this, my first solution was, okay, we should gather really all the different experts in the field that are necessary to study these nutritional and metabolic properties. So first, process engineering colleagues that were able to develop processes to concentrate buttermilk and butter serum to provide a milk polar lipid-rich ingredient, rich in milk fat globule membrane. Other experts that were able to design dairy products and measure the functional properties of these butter serum concentrate and milk polar lipids. Colleagues experts in lipidomics and sphingolipidomics in order to refine our knowledge of the composition of these milk polar lipids. And of course, collaborators that are experts in human lipid metabolism, fasting and postprandial, microbiologists, expert of the gut microbiota. And by putting all these people all together, we could set up a processing to obtain an ingredient that was sufficiently rich in polar lipids to be studied in high dose in humans. We could characterize the fine molecular composition of sphingolipids in these buttermilks and ingredients and their functional properties, and elaborate an isonutrient dairy matrix that was a cream cheese, 3 different cream cheese that had exactly the same macronutrient composition, but with different proportion of milk polar lipids therein, that could allow us to study this in a clinical trial in overweight postmenopausal women in order to evaluate the potential metabolic benefits. And we studied 3 different doses in order also to envision a potential risk-benefit effect. For example, if we had seen interesting effects at 3 grams, but maybe a potential problem at 5 grams. So this was sufficiently high dose to envision benefits, but also potential risks. And finally, we could also combine different clinical approach to really try to understand both the metabolic effects of these milk polar lipids in cream cheese and some mechanisms. So there again, there was here a clinical trial where after 4 weeks of daily intake of this cream cheese, we could observe benefits on lipid metabolism altogether, but also benefits on sphingolipids with a decrease of some pro-inflammatory sphingolipid species. So this was really a demonstration of the chronic intake for 1 month with beneficial effects of the milk fat globule membrane supplementation. But to understand also what could be the mechanism during the postprandial phase, we also elaborated a complementary clinical trial in ileostomized patients, additional also to preclinical studies. Ileostomized patients have a pouch at the end of their ileum. Therefore, it is possible to collect everything that exits the small intestine. So by analyzing this efflux, we could, and with also using stable isotope tracers, here again, we could conclude that, in fact, the beneficial effect of polar lipids on lipid metabolism could be due to the fact that there is an increased efflux of cholesterol and milk sphingomyelin at the exit of the small intestine. It means all cholesterol and sphingomyelin are not absorbed by the small intestine, but a significant portion of them are just exiting to the colon. And we could further show here, potentially in the postmenopausal women, a role of the gut microbiota, because there was also a fecal increase in coprostanol, that is a metabolite derived from cholesterol by the action of some specific bacteria of the gut microbiota. And in the literature, increased fecal coprostanol is described to be associated with benefits on lipid metabolism. So altogether, our combined approach shows that we could show that milk polar lipids can trap cholesterol, take it down to the colon where it can increase fecal coprostanol, and also there is an association with increased sphingolipids from the milk polar lipids. So it is really this association of different clinical models, the use of tracers, and also fine lipidomic techniques together with the metabolite of the gut microbiota that could allow to suggest potential mechanisms. So really in the field, the future direction are really today to the identification and the understanding of the dairy matrix effect in nutrition and health, especially via the specific properties of the fat globules and structures in the dairy products. And this is relevant really at all ages, from young to elderly, and also in disabled and diseased persons. So I think we can discuss this later in the discussion. Also, this highlights currently that milk lipidomics is very important, and minor lipids are maybe not so minor considering their potential major effects in metabolism of the gut microbiota. And so interactions of the gut lipids with the gut microbiota is also a very hot topic today. And there is also a challenge maybe of the community to recapitulate the data from cohort epidemiological studies and randomized control trial data regarding the link between milk lipids in dairy and health. Because also, of course, the dairy are within complex dietary pattern, and there are international variability in the intake. So these are really interesting future directions in the field. And so my personal conclusion would be that it's always most useful and exciting and fruitful to really always try to combine, associate different disciplines, technique, approaches, integrate new concepts sometimes from other fields, and interact as much as possible with colleagues with complementary expertise. Also that when working on product processing and formulation, and there are always new trends in the field, always envision what will be the consequences regarding the fate in the digestive tract and metabolic impacts of these differential processings. And also there are, so many ways to further explore how milk lipids and fat globules will contribute to the dairy matrix effect. And the next speakers will envision all these aspects of the very passionate about milk fat globule secretion, the digestion, and a new application in innovative nutritional application. So let's do it. And I will be glad to contribute to the discussion at the end of this webinar. And of course, many acknowledgements for me to all my team and colleagues and funders. Thank you for your attention.
Merci, merveilleux, superbe. This was wonderful, Marie-Caroline.
Thank you.
Okay, let me remind the audience, not only do we have the great joy of listening to these 4 scientists describe their research, we get the added bonus, we can spend an hour asking them any questions we would like. So in the Q&A section of the Zoom call, at any time, you can ask a question. And we will get to that in the discussion section at the end of this conference. All right, it's going to be very difficult to top that, but I am delighted to invite Nurit Argaman to present her brilliant research. Nurit.
Thank you, Bruce. So being active in dairy lipids for quite a while now, I also understood the necessity of delivering appropriate alternatives. So this is my disclosure. I co-founded these 2 companies. Oops. Sorry. Okay. So as I indicated, I am interested in milk lipids for quite a while now, and especially in their unique properties. So as Dr. Michalski explained very vividly before me, milk fat is secreted in a unique structure termed milk fat globule, and it's unique due to several different aspects. The most interesting ones for me, at least, are the fact that this particle is enveloped, this triglyceride droplet is enveloped by 3 layers of polar lipids, where usually we find that polar lipid particles are emulsified by a single layer of polar lipids, but the mammary gland utilized 3 different layers to basically envelop this particle and emulsify it in the aqueous phase, which is milk. And another very unique property is the size of diversity of these particles, which in many different mammals, basically being secreted in a wide range of sizes from less than 1 micron to over 10 microns. And this diversity is maintained throughout evolution of mammals. So the fact that some pressure maintained variability suggests that there is some kind of a benefit to this size diversity, either to the lactating mother or to her infant. And we were very intrigued by these different aspects of milk lipids, their unique properties. And what I've been, my aim over the years were basically studying the aspects of lactation. So what happens inside the mammary gland that regulates these structures that eventually secrete this very unique structure in a very unique composition and in a specific yield. So what are the intramammary mechanisms which control these properties of milk fat? What are the end consumer can benefit from these changes in concentration, composition, and structure? And what are the environmental and nutritional impacts of the mechanisms that take place in the mammary gland and result in the formation and secretion of milk lipids? And what I'm going to do today, I'm going to basically focus in each of these aspects and show you what are the challenges that we're facing when we're asking these questions about these processes, and how did we get our way around these challenges and hurdles, and basically being able to answer our questions and establish our hypothesis. So we'll start with the environmental, there's some nutritional impact on milk lipid composition. So it all started with reports from our group and others as well that milk produced by dairy animals have better quality than milk produced under confined management, under confined conditions. And what we were interested to know is if it's indeed the diet that improve milk composition of goats, dairy animals. In this specific project, we used dairy goats. So is it the diet that the goats are fed or ingested when they graze, or is it something else? So you can think of maybe social interaction when going out to the pasture area, or physical activity even, which is completely different between goats that actually graze compared to goats that stayed indoor and fed their diet. So can we really pinpoint the specific dietary component that is responsible for the change in the lipid composition of the milk? What we did, we had 3 different groups of dairy goats that were fed hay-based diet. This is the commercial diet used in Israel to feed dairy goats. We had goats that went on to pasture. They went on grazing, so they can collect their own forages. And goats that stayed indoors under confined conditions and were fed the forages that were collected in the pasture area. So basically we had these two groups, the pasture and the hay, that stayed under the same conditions but fed different forages. And we had the pasture group and the pistachia group. Pistachia is the main forage that is found in the Israeli grazing area. So these two groups had similar forage intake, but different feeding conditions. And what we saw is, especially in terms of their tannin content in their diet, tannin are secondary metabolites produced by forages, by the vegetation in the pasture area. The tannin content was very low at the hay-based diet, the commercial diet, and was relatively pretty high actually in the two other groups. So regardless if they consume these forages in the grazing area or indoors in the dairy barn, they had very high content in their diet of these tannin components of the diet. Their dry matter intake was similar, their protein intake was similar. This was actually one of the only differences between these three diets. What we saw is that maybe was expected. We saw that milk yield was similar between the groups, but milk fat was much higher in the groups that consumed the tannin-rich diet, and the polyunsaturated fatty acid concentration was also altered by these diets with higher omega-6, but mostly higher omega-3 fatty acids in the milk that produced by the dairy animals, by the dairy goats that consume tannin-rich diets. What is the source of these lipids? We did not know, because when we looked at the fatty acid composition of these forages that were consumed by the pasture groups, we did not find any polyunsaturated fatty acid at all. And what we were hypothesizing is that it's not the origin of the fatty acids from the feed that resulted in elevated concentration of polyunsaturated fatty acids in milk. It is the tannin capacity to basically reduce stress in the animal as a whole, and specifically in the mammary gland, and therefore enabling to use these polyunsaturated fatty acids to secrete as a milk component, rather than using them as a precursor for different molecules that can control stress response of the cells. And if you're asking yourself what kind of stress these animals are facing, so in the mammary gland, lactation is a metabolic stress in itself. So there are a lot of pro-inflammatory processes that occur in the mammary gland that basically control the milk synthesis and secretion processes. And we hypothesize that these tannins are actually exerting some kind of modulation of the stress in the mammary gland. But the challenge was how to prove that these tannins are actually delivered from the GI tract to the plasma, and from there to the mammary gland itself. So just a quick note, tannins is a huge group of chemicals. Some of them we know, the part of them we know as polyphenols, but it's not all the group of tannins. And this group contains chemicals that are that have a variety of compositions and structures. Some of these tannins are actually never absorbed from the GI tract into the blood. Some of them are absorbed, but after significant chemical alterations and modulations due to the enzymes and actual epithelial cells of the gut. So it's very hard to mimic the composition of these secondary metabolites from the plants that are actually absorbed into the bloodstream. And what we were trying to understand is how can we prove that these molecules actually absorb to some extent and in some composition into the blood, and exert also a systemic effect that can directly impact the mammary gland lactation process, and not only exert activity in the GI tract in itself. So actually understanding or analyzing the blood and characterizing exactly what is absorbed and in which exact composition. This is a project that our colleagues expected that it will take a couple of years. So although we're going this path as well, we were trying to achieve more rapid results. We were very interested to see if indeed these polyphenols, these tannins, exert some kind of a systemic effect. Can they reach the mammary gland cells and impact those cells directly? So we looked for a faster model that will help us to answer this question. So here's what we did. We actually used goats that as donors for blood. So these goats were either fed under confined conditions, this hay-based diet, which has very low content of tannins, or goats that went to the grazing area and collected their own forages that have high tannin content. So these goats were used as donors. We collected blood, not a lot, 50 mLs, and separated the plasma. And this plasma was used as a conditioning media for mammary epithelial cells grown in culture conditions. So we used the plasma to basically feed those cells and looked at what their lipogenic capacity and how they deal with stress. What we found is that actually we were able to mimic some of the unique properties of the goats that went on pasture in the in vivo study. And these cells, when they were fed the plasma, or they were treated with the plasma of goats that were fed on high tannin diet, had a higher lipogenic capacity. And supporting the hypothesis that I showed you earlier, these cells that were treated with the plasma of the grazing goats had a higher capacity to maintain production under stress conditions. So here you can see results from seahorse system that use different toxins to really stress cells. And it monitors their ATP production capacity, their oxygen consumption capacity on real-time experiments. And what you see that all of these parameters that basically suggest that these cells are more resilient to stress were much improved by the plasma of the grazing goats compared to the plasma of the goats under confined experimental conditions. So the results here, basically, first of all, what it told me as a scientist, that this is the ability to be able to perform experiments in different models, in vivo, in vitro, and ex vivo model, really can leverage on our understanding in general, in the lactation processes that takes place in the mammary gland, and also specifically the lipid metabolism processes in the mammary gland cells. And also specifically for this experiment, we found out that the polyphenols or the tannins maintain their post-absorption activity, and they basically provide mammary epithelial cells protection against stress conditions, which we believe induce the ability to use omega-3 fatty acids specifically, but in general polyunsaturated fatty acids as milk, and not, these fatty acids can be secreted into milk and not used by internal metabolism of the mammary epithelial cells themselves. Next, I want to share with you another very interesting aspect that is studied in my group is the intramammary conditions that may or may not affect fatty acid composition and the macrostructure of the milk fat globules. So as I told you before, we were very interested in understanding what is the regulation mechanisms, if there are regulation mechanisms that basically modulate the milk fat globule size. So we know that milk fat globules, and this is actually a paper that was published a while ago from Marika Rolin herself, showing that the part of the parameters that control milk fat globule size is the time from birth, time in lactation, in the lactation period. Other factors that may impact milk fat globule size are, as I show you, diet, and of course interspecies differences. But eventually, all mammals have high variability in the sizes of the milk fat globules that are secreted into milk. And we are very intrigued by these specific characteristics of milk fat. And we are trying to understand what is the implication of this ability to synthesize small or large milk fat globules. The first thing that the first population that we were working on for several years now are bacteria. Bacteria that basically resides or populate the cavities, the body cavities in the mammary gland itself. So the mammary gland has a structure of cells that synthesize and secrete the milk, and this milk is secreted into alveoli lumen and transported through ducts that eventually transport the milk to the consumer, to the outer environment through the teat canal. So these are body cavities. All these transport mechanisms are body cavities eventually that are exposed to the external environment, and therefore there are bacteria coming from the external environment and penetrating basically these ducts and alveoli lumen that and there it interacts with milk components. They are exposed to milk components. And again, we were interested in understanding what is the function of size in this interaction between bacteria, again, populating the mammary gland itself. And we wanted to do that without causing a real damage to the animal, because understanding this interaction using in vivo model can cause a significant damage to the animal itself. And if you're trying to move that into in vitro model, where bacteria and cells are co-cultured, then you have very limited time frame to study this interaction, because the bacteria will outgrow the cells and kill them in a matter of several hours. So we're trying to work our way around this limitation, and we came up with this idea of this disintegrated udder model. And what we did is we took milk and separated the milk fat globules according to their sizes. So we had large and small milk fat globules, and we used them as a substrate for bacteria. In this specific experiment, we used Bacillus subtilis. It's a non-bacteria. It is very common in dairy plants, in dairy barns as well. And it was also shown several times to be part of the udder microbiome. So it is present in the udder itself, in the milk collecting ducts. So it's very relevant in terms of the interaction in the mammary gland itself between milk and bacteria. So again, substrate were either small or large milk fat globules. And after incubation with the bacteria, we collected the soup of the bacteria, and we ran several analyses. What are the metabolic output of this bacteria? And we could also use this soup to run some functional assays, which I'll share with you in a minute. And we could also, of course, follow the growth trajectories of these bacteria during incubation and after incubation with the milk fat globules of different sizes. So the results were very convincing. Okay, so here you can see large milk fat globules or small milk fat globules on the right-hand side, stained in yellow-red stain. And the bacteria itself is has a green fluorescence protein marker. So as you can see, when the bacteria was incubated with small milk fat globules, it grew almost tenfold higher compared with its growth capacity when large milk fat globules are used as a substrate. However, large milk fat globules induced biofilm formation by these bacteria in a dose-dependent manner. Here you can see the different doses of the small milk fat globules that were mixed inside the substrate, and you can see how large milk fat globules resulted in the formation of very developed colonies compared to the small milk fat globules treatment. And of course, if small induced growth and large milk fat globules induced biofilm formation, the metabolic the metabolism of the bacteria is expected to be completely different. And here in this heat map, you can see how the metabolome of this bacteria was segregated or significantly distinguished between the state that it was in when grown on small or large milk-fed globule. So a complete segregation between these two stages. As I told you, this bacteria populate the mammary gland ducts themselves, and therefore it is directly associated or can directly exposed to the milk-fed globules that are secreted by the mammary gland. But not only that, since it is found in the alveoli lumen itself, the metabolome of this bacteria or its chemical fingerprint basically can affect the host cells themselves. Okay, so because of the co-localization of the bacteria, the milk, and the host epithelial cells. And we're very interested to understand if there is a different impact on the host cells, if the bacteria is metabolome is originating from growing bacteria or from a bacteria that is in a biofilm formation mode. So what we did, we took our in vitro culture of mammary epithelial cells isolated from a cow udder, and we primed it with LPS. So we induced a pro-inflammatory mode, and then we exposed the cells to the soup of the bacteria that was grown on small or large milk-fed globules. So you can see here in two colors the two different doses of LPS that was used, and I will ask you to look at the blue graph that you can see here the mRNA expression of these pro-inflammatory cytokines and how their expression was elevated by introduction to the LPS, and how when the soup of the bacteria grown on small but not large milk-fed globules basically reduce the pro-inflammatory response, whereas the soup of the bacteria grown on large milk-fed globule really enhance the pro-inflammatory response. Now, whether it's good or bad, we don't know yet, but we do know that there is an impact on the host cells themselves in a cycle that the bacteria respond to the milk-fed globule size by a different growth mode and release different metabolome to the environment, and the host cells themselves which are exposed to this metabolome also react differently and can really change their capacity to deal with stress, this time activated by LPS. So we were very intrigued by these results, and what we were also trying to figure out if this is a universal impact. So the size of the milk-fed globule will change growth trajectories on any bacteria used. And here you can see 3 different strains that were isolated from the udder of a mammary gland. And you can see how the subtilis and the plantarum basically respond to the size of the milk-fed globule used as a substrate, but E. coli was not. So it didn't really respond to the different sizes. So we were asking ourselves, if there is any kind of another role played by the milk-fed globule size that can change the response of the bacteria or the interaction of the host with the bacteria. Here, again, to answer this question, we used mammary epithelial cells in culture that were fed different treatments and induced to produce large or small milk-fed globules. And once this was achieved, they were co-incubated for 2 hours with E. coli isolated from cows with mastitis. So we knew that this is the strain that is pathogenic. And after 2 hours, we washed the medium away, we collected the cells and let the bacteria grow and monitored their number using OD of 600 nanometers in order to quantify the bacteria that were actually either adhered to the cells or penetrated the cells when they secreted small or large milk fat globules. And what you can see here is that the cells that secreted small milk fat globules had lower bacteria count, which means that by secreting small milk fat globules, the cells have some kind of a protection against adherent of pathogenic bacteria or even penetration of pathogenic bacteria. So we were able to basically monitor the interaction between the bacteria and the milk fat globules without killing any dairy animal and without introducing a very destructive environment to our mammary epithelial cells model. So the last thing I want to show you is the impact of the milk fat globule structure on the consumer, and this time on the consumer can be the bacteria in our gut or the consumer itself in let's say in vivo model of milk consumption. The first thing that we wanted to understand, and this goes, this basically kind of similar to what Marie-Caroline was asking in her studies. We were wondering about the implication of, or we wanted to distinguish between the implication of the chemical composition of milk fat and distinguish it from the structure. So basically we're asking a question, is the structure in itself has some kind of importance for the physiological response for the consumption of dairy fat? In order to answer that, this was the challenge of how to distinguish between the chemical composition effect and the structure effect of the milk fat globule, again, due to the fact that this has a very unique macrostructure that cannot be found in any other basically dairy or fat or lipid source in our diet. So the first experiment that we ran was on bacteria. We basically formulated a lipid mixture that had a similar composition of polar lipids and triglycerides as the large and the small milk fat globules. However, it did not have the structure. Okay, we used streams of the dairy industry, so we had basically similar composition ratio between triglycerides and polar lipids, but these assembled milk fat globules, these formulated milk fat globules, did not have the same structure. And what we saw again when we used that as a substrate for our Bacillus subtilis model bacteria, we saw again that the small milk fat globules really enhanced the growth of the bacteria compared with the large milk fat globules. However, the formulated small or large milk fat globules, although they had similar chemical composition, did not manage to biomimic the enhanced growth or the induction of the growth of the bacteria as the small native milk fat globules with this natural structure. So maybe evidence number one of the importance of structure for the consumer, this time the consumer is basically the bacteria. We also wanted to take it one step further to study the impact on in vivo model in a preclinical study. And again, we wanted to understand the impact of structure and to try to segregate between the structure and the composition effect. Here we used rats that were fed high-fat, low-protein diet compared to the control diet. And this high-fat, low-protein diet had either the optimal content of micronutrients, similar to the control diet, or it was micronutrient deficient diet. So very harsh deficient diet for these young rats. And we used different commercial dairy products to basically change the structure of the dairy fat that was used. So the fat in this diet was either soy oil or 3 different dairy products, cream, which is the only liquid dairy product in Israel that where the fat is not homogenized, so the milk fat globules are found in cream in their native structure. When this cream is churned and turned into butter, it loses the structure, but some of the polar lipids, some of the membranes stayed trapped within the crystalline form by the churning process between the triglyceride molecules. And ghee is a purified butter that basically lose all the polar lipids and proteins that are in the membrane and maintain only the triglycerides themselves. So very similar composition, fatty acid composition between the three products, but very different structures. I will show you two very short examples of our results. The first one is the length of these animals at the end of the experiment, 7 weeks of experiment. You can see how the high-fat diet with the sufficient amount of micronutrients really reduced the growth capacity of these animals. This was the results both in the high fat with no micronutrient deficiency or with micronutrient deficiency. And you can see how the cream, basically, that was provided to them as the fat source in their diet, really compensated for the scarcity of protein in their diet and even micronutrient deficiency. So the growth was almost not affected at all by the high fat, low protein, low micronutrient diet when cream was used as a fat source. It was obviously not a fact with the ghee, that the purified butter, that was actually resulted in the worst growth results, and also not so much with the butter. So very clear indication of the importance of structure. I want to also show you something that may say a slightly different result or slightly different conclusion. Here you can see some of the bone parameters that were taken by micro-CT, and you can see the control here in red. And what you can see is when we use the micronutrient deficient diets, how butter and cream to some extent compensated for the loss of micronutrients and the loss of protein in the diet or the reduced protein content in the diet. However, ghee and oil resulted in a very bad structure of the bone that was not supporting neither strength of the bone nor flexibility of the bone. So to conclude my work, I think we can definitely say that milk fat macrostructure, first of all, is modulated by internal signals and environmental conditions that the animal goes through. And this sensitivity basically can be used to modulate milk composition and structure, which can then affect digestibility and bioavailability of lipid constituents. Industry should acknowledge the importance of the macrostructure and its benefits, and I think this is the same statement that I really appreciated that Marie-Caroline before me had. And most importantly, I think nowadays is the fact that alternative sources of fat, which aim to biomimic milk lipids, should not be limited to the replication of the chemical composition, but should aim to replicate also the structure. So with that, I want to thank the students that were part of all of the experiments that I shared with you today, and our collaborators, and thank you for the invitation.
That was magnificent. I have so many questions, but I will save them for the discussion period later. Thank you very much. It's now my great pleasure to introduce a remarkable scientist and a truly global scientist, Ben Boyd, pioneering some of the most remarkable research on milk lipids ever. Mesmerizing. Ben, take it away.
Thanks, Bruce. Thank you, Jennifer, and Alika also for the organisation and invitation to speak at the meeting. It's a great initiative, and I'm just backtracking my slides here so you get a quick preview. So I have this joint appointment between Denmark and Monash University. Most of what I'll show you is work that we've done at Monash, where we're very interested in structure in lipid systems. So Narita's really given a great push towards the structure theme, and I'm certainly going to continue along those lines. So here's my disclosures, and none of the data that I'll show today comes out of any of those disclosures. So so let's start here. So we've already heard a lot about the milk fat globular membrane from Murray Caroline and from Nuri. And so a couple of little points here. So the milk fat globular membrane, it's super interesting and has a lot of function built in with protein content and different lipids. If we're interested in lipids in general, and the title of the session is Milk Lipids, not MFGM. And so we need to kind of recognise and acknowledge that of all of the lipid that makes up milk fat droplets and milk fat as a component of milk, only or less than 2% of that is associated with the milk fat globular membrane. More than 98% of that lipid content is associated with the mostly triglyceride form. So if we want to really come to grips with what's happening with milk lipids, we shouldn't be ignoring the fact that more than 98% of the lipid is associated with the core and not with the globular membrane, as important as it is. And so surely we want to know a bit more about that. And that's kind of the theme for the next 15 minutes or so. So challenge one that I've put out there was, what actually happens to milk lipids when we consume them, and I'm talking primarily or globally at least. So that behaviour is likely to be dominated by the behaviour of the triglycerides in the core of the milk, the milk fat droplets. And so, but I'd also say, well, shouldn't we know? So we've studied milk for quite some time in science, and I think it's reasonable to say that we're still coming to grips with what actually happens to lipids during the process of digestion. And so this is sort of captured in summary or in schematic, if you like, here, where when we ingest lipids, and if you're not particularly familiar with lipids and how they are processed by the body, then very, very simple terms here, lipases will digest the triglyceride, and we can see in the text on the arrow here that I've deliberately used small font for lingual lipase, so there's very little activity in lingual lipase, a small amount towards lipids in the oral cavity. There's a moderate amount in the stomach, so small or moderate amount of gastric lipolysis occurs. Most of the lipolysis occurs in the small intestine under the action of pancreatic lipase. And so pancreatic lipase's job is really to cleave the two fatty acids from the outside of the triglyceride molecule to liberate a monoglyceride, the two fatty acids. And at least in a sort of a blind nutritional context, if you like, those units then are small enough to be absorbable. So the intact triglyceride and intact diglycerides don't, or at least in the adult gut, are not absorbed intact in any significant quantities. So our body has this lipolysis process specifically there to consume quite a lot of fat. If you think about, if you eat 2 Big Macs and a large fries, then all of that fat is quantitatively broken down into monoglycerides and 2 fatty acids and absorbed by the body. And that has obviously unfortunate consequences if that's your diet. But that's what happens, and then it's reassembled on the other side back into triglycerides for transport around the body. And so these, or primarily, and so these components then are more polar lipids than the starting triglyceride. And so I'm a physical chemist by training, and so my physical chemistry training would say, well, we've got polar lipids that are almost like surfactants, then we would expect them to self-assemble in an aqueous environment and form potentially some interesting structures. And so I would have anticipated that we would form different colloidal structures, and it's reasonably well understood that and micelles and bilayer structures probably do form in the gut. But there's also potential to form other types of structures. So these more highly ordered self-assembled structures. And so the one on the left here is called the cubic phase. The one on the right-hand side here is an inverted hexagonal phase. These structures are no stranger to physical chemists that are working in the lipid area, but possibly are less familiar to many of the people on the call. But their primary feature is that there's water channels that permeate through these structures. And so if during the digestion of a milk fat droplet, that the composition is such that we have enough polar lipids there, and if they've got the right properties in terms of chain lengths and geometry, and I'll talk a bit more about that in a moment, then we would anticipate that they're going to form these self-assembled structures during digestion. So how do we then go about finding these structures or looking at these structures? And so so back in the 1970s, you would have just taken, you're a smart guy, you'd have taken your microscope, exposed an olive oil droplet to some lipase, and observed that you get some interesting material forming at the interface of the fat droplet with the aqueous phase, and then you write a science paper, and it's all good. Now that's great, but it doesn't tell us a whole lot about the nature of those structures or give us any of the sort of finer detail of what those particular structures are that are forming in the interface. So if we fast forward a little bit, this is a in vitro lipolysis model now. So rather than doing this in essentially a sort of constrained solution environment, now we can control the pH or monitor the pH at least. And I won't elaborate too much on this model other than to say that it's effectively like an artificial in vitro gut sort of model. And in the intestinal context, then we can introduce some pancreatic lipase, bile salts, etc., and see what structures are actually formed during digestion. So in this sort of late 90s, early 2000s period, there were some studies using this sort of an approach, and usually they would be coupled with something like cryo-TEM to try to look at the colloidal structures that are formed during digestion, or they might actually use what we call small angle X-ray scattering. And so just going back to when we have these ordered structures, if you shoot X-rays into these sorts of systems, then if there's order within those droplets, then they'll scatter X-rays at a characteristic angle. And so you can get what we call diffraction peaks at characteristic angles that should tell us what structures are actually there during the digestion process. But the problem is that by the time you take your digesting system and then you maybe separate an aqueous, the aqueous component from the lipid component, or even just transfer it into a small angle X-ray scattering instrument, it's already hours or potentially even days that have elapsed. So you lose all of the ability to really look at properly at time resolved behaviour, and it induces a high level of artifacts. So it was in the sort of later 2000s and around 2010 that we actually took this model, but now rather than using a low-powered X-ray source that you would typically find in a lab, we could couple it up to a synchrotron source, and that gives very high flux and high intensity X-rays that we can then use to actually probe what's going on inside those lipid droplets during the digestion process in situ. And so if you do that with milk, then what you actually see is something like this. So we start with a more or less featureless diffraction pattern because there's no ordered structures at the start. We've just got liquid triglyceride in there. And as you can see, I'll let this video run, then we see some transformations between from the unstructured droplets through to, we start to see diffraction peaks that are characteristic of different self-assembled structures inside those droplets. So as the lipid composition is changing to more and more polar lipids, we're forming different types of structures. And it turns out that we actually end up at the end of the digestion process with what we call the cubic phase, the bicontinuous cubic phase structure. And from a physical chemistry perspective, if we look at this process, then it actually does make a lot of sense. So this is now the video turned into a waterfall profile. So we've got our diffraction angle along here, and we've got time of digestion going backwards here. And the sort of order of procession, if you like, of the generation of the different structures is what we would have expected from the theory called critical packing parameter, which really talks about the geometry of these molecules, or the global average geometry of the lipids that are present, and the fact that you should see specific structures forming in a specific order, and that's exactly what we saw during the digestion of bovine milk. So I showed this to Bruce, and then he was quite excited. So we've done some other things since then. So challenge 2 that I'm sort of putting out there is, so now that so if I go back, so to kind of summarise solution 1, if you like, we see these really interesting, you can see these really interesting structures that are formed by milk lipids during digestion. And so that's, that is essentially a feature of milk, and I'll talk a little bit more about what I mean by that. And so now that structure, well, now that these, the fat component of milk lipids, and not just the membrane, but the internal core, and have also really interesting structural characteristics and evolutions of structure during digestion, then it's reasonable to ask the question of, well, could we actually use structure rather than composition as the actual discriminator between different systems? So if milk forms particular structures and other systems form different sorts of structures, then maybe that's actually an interesting signature and an interesting difference between them. So we're usually talking about composition in lipid systems rather than structure. So that's kind of the theme of the next challenge, if you like. So if you take milk and then you compare milk, and I'll talk about milk in the most general sense, to other types of systems where they may be considered as a substitute for milk. So we've got some plant-based products on the right here and some infant formulas on the left. Do they then behave similarly in a structure sense to milk, or is it actually a useful discriminator between systems? So if we look at some other milk, so some other mammal milks, then on the left-hand side here, we've got goat milk. And so it turns out that goat milk behaves very much like bovine milk during digestion. So we see different structures forming. So there's cubic phase, hexagonal phase, plus bicontinuous cubic phase. You don't need to remember the exact structures of those, other than to say that, at least as a fingerprint, the structural behaviour of goat milk during digestion is very similar to bovine. It's not exactly the same, but that kind of probably makes some sense. And then if we look at human breast milk, well, that actually behaves slightly differently under exactly the same conditions. If we digest the same fat content of milk under the same conditions, and in this case we see we've got this formation of inverse micellar cubic phase. So that's this representation down here. Again, don't be too obsessed with what the actual structure means, but it is actually signature of human breast milk that this that towards the end of digestion that this structure is formed. And so the takeaway at this point is, okay, we've looked at 3 different mammal milks, and they all form highly ordered self-assembled structures during digestion. So the question then is, so what about some of those other systems? I'm waiting for my slides to advance here. Right. So if we then look at some of the so-called plant drinks, I'll call them plant drinks, soy drink in this case. So if we digest soy drink under exactly the same conditions as bovine milk, and it's a specific brand of soy drink, I won't elaborate here, then it's quite apparent that it doesn't behave in the same way. So it doesn't form those highly ordered structures that we've seen in the digestion of bovine milk. It's essentially a featureless profile, indicating that the lipids are not amenable to forming the same types of structures that all of the mammal milks seem to do. And if we repeat that experiment with almond milk and rice, almond drink, I should say, and rice drink, and compare that to mammal milks. And we can see it behaves very similar to the soy extract, but not similarly to any of the mammal systems. So there is a clear structure-based discriminator there between the mammal milks on the one hand and the vegetable-based drinks on the other hand. That's obviously a teaser into question time, so feel free to ask me the obvious question then. So what happens in the in between? So we've got we've seen what happens with those with bovine mammal milks and the vegetable milks. So infant formula, I guess we can say, is somewhere in between. So again, I've got 4 different infant formulas here, and I'm not going to elaborate on what they are. You know, the marketing around infant formula is quite interesting. So some of them claim to have learned from the breast, but I'd like to know what they learned, because they don't know that human breast milk forms an inverse micellar cubic phase. But what we find is that infant formula 1 actually behaves at a structural basis very similarly to human breast milk. 2 forms an ordered liquid crystal structure, but it's different to human breast milk. And then 3 and 4 behave very much like the other vegetable drinks that were showed on the last slide. And it turns out, if we've done the lipid analysis on these, it turns out that that would have been anticipated. This one appears to contain more milk lipids than the others do. So there's a clear opportunity here to use this structured formation during digestion as a tool to discriminate between the different systems. Then challenge 3 is around, well, if we now use structure as if structure is a feature of these systems that we can then potentially use and exploit, then can we design systems from a structure perspective rather than composition perspective, and then what's the potential for implications of that? So Andrew Lulo was a postdoc in my group at Monash up until very recently. So he was asking this question of, if you take, so we know that milk, and human breast milk in particular, is extremely complex because of the potential combinations of the different fatty acids on triglycerides. Is it possible for us to take homotriglycerides, where all the fatty acids are the same, and mix those together and generate a composition that behaves structurally the same as human breast milk, even though the composition is quite different? And so it turns out that when he asked the question of how complex does a mixture need to be, the magic number is 7. So you take 7 homotriglycerides with the right fatty acid chain length, and you put them together in the right combinations, or right proportions, I should say, then they will form a complex mixture, or much less complex mixture, but that behaves structurally the same as human breast milk during digestion. So if you take 6, 5, or 4, then you don't have the same sort of behaviour. You need 7 and in the right proportion. So if we then extend that to something like colostrum, so Shazza was a PhD student very recently. She's participated in a couple of IMGC conferences. So she's made a colostrum substitute using a similar kind of approach, and has incorporated some protein and HMOs into her substitute system and compared that directly to colostrum. And so it turns out, or at least to human breast milk, so it turns out that if you take, this is a similar approach to Narit showed towards the end of her presentation. So if you culture some intestinal cells with some health promoting and anti-inflammatory promoting bacteria, and then you insult those cells with LPS, then if you treat those cells also with infant formula, human breast milk, or the substitute, then what she found is that the controls, so the LPS, so negative control, if you like, is very detrimental to the cells. If she uses infant formula, then there's at least in the presence of the bifidobacteria, performs better than just LPS on its own. That makes sense. And then if you have human breast milk or the substitute, then you see a much better recovery of the cells in that case. after insult with LPS. So there is a potential. Well, I haven't linked it deliberately. Haven't linked directly to structure here, other than to say that we know that lipids now self-assemble during the digestion process and form some interesting structures, and they can then be used to discriminate between different types of systems. And so the question then is, is linking that behaviour then back to potential benefits in terms of performance. So future directions. So we still need to ask, why is there a discrimination between mammal and vegetable sources? So we know there's a difference there, but is there a relevance in that difference in terms of, or is it just a coincidence that yes, the lipid compositions are kind of clustered. We've done some lipid clustering using PCA and seen that that's important, but is there some kind of higher reason for that? We also know separately that structure affects drug absorption. We need to gather evidence on the nutritional impact of that. And then lastly, there's it kind of echoes, I guess, Narit's comment that, and Marie Caroline, that structure is an important factor. It's not just all about composition. And so hopefully I've convinced you once again, as well as the first two speakers, that that's the case. So thanks for your attention, and back over to you, Bruce.
Magnificent as always. I am always impressed with what the techniques that you bring to this science can show us. All right, it's also killing me not to ask you questions. I'm holding off for the discussion. All right, and last, but certainly not least, one of the most inspiring young scientists in the field of neurobiology and the implications of diet to neurological health, Amir Taha. Amir, take it away.
Thank you, Bruce. Thank you all for being here. I am honored to be invited and to present some of our recent work on milk. So I'm going to talk today about milk bioactives during whey production and how they affect brain function. I assume everybody can hear me okay. So disclosure, I'm clean, nothing to disclose except for my funding from the NDC, USDA, and Alzheimer's, at least related to this talk. So a bit of background, the cheese making process is complicated. So if you think of milk, the proteins in milk broadly and simplistically speaking, there's the casein fraction and the whey fraction. The casein is what is used to make cheese, and the whey is, in the past, it used to be a byproduct or even a waste product. But then humans realized that we can use it to build nice muscles, which I lack, obviously. So then we developed ways to fractionate it through filtration to make whey protein isolates and whey protein concentrate, WPC, and whey protein isolate. And I'll get to the bottom part of this chart in a few slides. But one of the new byproducts of this process is something called WPPC, which is whey protein phospholipid concentrate. So just like the name tells you, it's more concentrated in phospholipids compared to other types of other milk fractions, such as the buttermilk cream and so forth, and the original milk. And that's why it's called WPPC. Our aim was to understand whether we could actually use this byproduct for brain health, and I'll explain why in a second. It starts with the observation that yes, it's high in phospholipids, and so is the brain. Actually, 80% of your brain lipids are made of phospholipids. The brain doesn't have much triglycerides. It doesn't have, which are found in fat tissue. Doesn't have a lot of cholesterol esters, which what your doctors would profile to give you the bad news that your LDL is high. It's mainly phospholipids. It's very lean in a way. It's 80% phospholipids, and the rest is actually cholesterol in the brain, in the free form. But WPPC is quite heterogeneous in composition. So it has, don't worry about the words, but it has phosphatidylcholine, serine, inositol, ethanolamine, just like the brain. In fact, if you look at, it's really weird. When I looked at this pie chart, I thought, wow, this is a brain sample, but it's actually a WPPC sample. So the brain does have roughly 30% PE, 30% PC, and it has sphingolipids for myelination, PSPI. So there's something to the diversity of the WPPC and its enrichment in phospholipids that we saw parallel the brain. And we thought, hey, let's have the two meet and see what happens. One point I want to emphasize: as much as there's a lot of phospholipids, as much as the brain is made of phospholipids, the brain phospholipids do not cross the blood-brain barrier. So you're thinking, okay, why am I giving this supplement to animals, as I will show you, and how is it getting to the brain? The mechanism is probably going to involve releasing specific fatty acids in these phospholipids. So if you look at the structure to the left, this is a phospholipid. These hydrophobic tails are actually fatty acids. So what happens in the blood is that you would probably release these fatty acids, and these would go into the brain, and the brain would reassemble them to make the phospholipids at once. And that's only because the brain doesn't trust anybody but itself. It doesn't trust the plasma. It doesn't trust any part of your body. It says, I'm going to use what part of the structure, the fatty acids, and what you provide, and I'm going to assemble these phospholipids on my own. Oops. Okay. So before I get into the brain story on what these things are doing to the brain, let's talk a little bit about one of the challenges for making such products. So one is product consistency during the scale-up. It's very variable, so making it difficult to always meet standard specifications. That's a challenge because if you want to take omega-3s, for example, to lower your triglycerides, the recommendation, I believe, is 3 grams per day. You need 3 grams. You want to make sure that your supplement has 3 grams in a day so you can lower your triglycerides. So it's the same idea. If this is being sold and the composition of phospholipids from one supplier is 2%, 5%, or within the same supplier, it varies by season, that's a problem. And this is critical because now there are standards for what WPPC should contain, the whey protein phospholipid concentrate. So as you see in this table, this is the composition that it should contain. So you can appreciate that the minimum phospholipid composition should be around 4%. And you want to get to that target. It'll be nice one day if we can have a range, maybe 4 to 10 or whatever. But for now, we have a lower threshold that we need to meet to call this a WPPC. And that target depends on the way you process things. Why? Because, again, when you milk protein-wise can be broadly and simplistically and in a reductionist manner, which my co-speakers are probably hate. You have the casein and the whey. Casein is used for the cheese. Whey is used for the whey protein. So you would filter the whey. Once you take out the casein from the milk, what's left is whey in solution, of course, the remaining milk fraction. You would filter it to get the whey you subject it through multiple filtration steps. The first one gives you the whey protein it gives you the whey protein concentrate, and then you filter that further to make whey protein isolate, which is more concentrated in protein and less lipids. So effectively, the lipids get filtered at this step between WPC and WPI to get the WPPC. So the byproduct is naturally rich in lipids because you're trying to remove the lipids. But where the processing, where you can introduce variability in the processing is at the WPC part, because sometimes you may want to get rid of the lactose and you would add a little bit more water. And in doing so, you dilute this and then you dilute everything downstream. So that's why there needs to be some consistency in the processing. So the solution basically is what we need to do is we need to go to one of the dairy processors and basically track how frequent, where, when do these inconsistencies occur? And this sort of stems from our own research, where when we measure these things in what's called WPPC, we do see a lot of variability. So I think the solution is to do more research to understand where things are, where the variability is really being introduced. All right, so challenge 2. So we move from this WPPC hopefully being consistent. Let's assume we achieve that one day. Can we use it to treat cognitive impairment? And well, for now, obviously, we need to start with preclinical animal models. So in this case, rodents, and then move on to humans. And part of the challenge with doing rodent studies, there's always a question of what's my dose? If I were to do a clinical study, what am I going to give people? And that's where we did a study to address these questions. A, does it prevent cognitive impairment? And B, at what dose or doses? And again, the reason we think WPPC can prevent cognitive decline, and I'll talk a little bit more about the model, where we induce it by high-fat Western diet, it's because it contains a lot of bioactive lipids, including phospholipids, which can be digested, as the prior speakers indicated. And then it goes into the plasma, and the plasma can release the fatty acids in these structures so that the brain can utilize them. Again, phospholipids don't cross the blood-brain barrier. They provide the substrates that the brain can use to reassemble the phospholipids it needs in the way it wants it. So solution 2, we performed dose response studies in preclinical model of cognitive decline, mimicking mild cognitive impairment in humans. So I work a lot on brain cognition and Alzheimer's, and I think the opportunity for intervention lies in this early stage of mild cognitive impairment, because in my view, at least, once you move on to other types to outright dementia, whether it's Alzheimer's, Lewy body, etc., it's probably too late. So this is an opportunity where we can sort of intervene and potentially stabilize or reverse or prevent. And that's the objective with testing this question in preclinical models. So what did we do? We used rats. We divided them into 4 groups. So as you see on the top left side, there was a low-fat control where the fat composition was 10% fat, as is the minimum needed for the rodent diet. There was a high-fat diet where the fat composition was 45%. And then there's the same high-fat diet, which we spiked with 1.6% of weight with the WPPC, and 10% of the weight with the WPPC. So 2 doses with the high-fat diet. And the high-fat diet is, it mimics sort of what a lot of us in the Western sphere are exposed to. It's high in fat, mainly saturated fats, and it's a model actually for people use it for studying diabetes and metabolic disorders, but people like me also use it to study cognition, because it's a model for mild cognitive impairment. We did cognitive tests at 2 months and at 4 months. And then we measured what's called the evoked response by implanting electrodes in the entorhinal cortex and the hippocampus. I'll get to that part of the story in challenge 3. But for now, let's focus on this part of the story. So what we did was we had control rats on the high-fat diet where we induced cognitive impairment, and rats on the high-fat diet containing 2 doses of the WPPC. And one of your questions probably is, what does 1.6% mean in human terms? And we think about these questions a lot, of course. Based on the calculus, and we don't have the human data at this point. We need to do it to obtain the evidence, but 1.6% would be equivalent to 1 tablespoon a day. So 10% would be equivalent to about 5 tablespoons a day. So at 2 months, so on the y-axis is the discrimination index, which is a marker of cognition. So higher discrimination index means you're more smart, so better memory. Lower discrimination index means bad memory. At 2 months, the numbers were all over the place. But at 4 months, the high-fat diet, so on the bottom x-axis, you can see the group stratifications. The high-fat diet did exactly what it's supposed to do. It reduced the discrimination index, or in other words, it impaired cognition. And what was even more exciting and interesting is that when the high-fat diet contained either doses of the WPPC at 1.6% or 10%, this cognitive impairment was reversed completely, as you can see. I'm not sure if you can see my mouse, but this is a high-fat diet compared to the low-fat. It impaired cognition, just like others have found on what it's supposed to do after 4 months. And at when the high-fat diet contained the WPPC at either dose, 1.6 or 10%, these impairments were prevented completely. So they look more like the low-fat group. At 2 months, again, we couldn't answer the question because there was no effect of the high-fat diet. We were trying to cheat our way to see if we can induce cognitive impairment earlier, but clearly that failed. So you need 4 months for this for the high-fat diet to have an effect. We sort of knew that, but we didn't, but we thought we should be adventurous. All right. This takes me to the last challenge. So hooray, we were able to prevent cognitive impairment that occurs with aging or with consuming bad diet. But knowing the type of, so you would say, let's go ahead and do a clinical study. And I or any clinician would tell you, great, let's do it, but who am I going to recruit? And the reason is because the causes of cognitive impairment in humans are so heterogeneous. So knowing the type of cognitive brain disorders that could benefit most from the WPPC allows you to recruit the right people. Because if you don't recruit the right people, then you get one of two outcomes. One, you get a huge positive effect because just by chance the right type of cognitively impaired individuals were in your study, or you get nothing because you just recruited a heterogeneous mixture of people. So the signal gets diminished. So I'll elaborate a little on what this means. Pardon me. There's many causes of cognitive impairment. It could be from, I don't know, playing sports and getting a concussion, for example, or traumatic injury. It could be from consuming a bad diet. It could be from diabetes. It could just be genetic. Environmental toxins. So there's many, such as air pollution. So there's many causes. And beyond that, the types of memories that could be impaired vary depending on the cause. So for example, vascular dementia, one of the risk factors for vascular dementia is high blood pressure, right? So, and that type of dementia, for instance, so you get patients who are cognitively impaired, they have MCI, mild cognitive impairment, but their impaired memory domain is more on the executive function than, say, the remembering stuff, what's called short-term memory, which is impaired in individuals with Alzheimer's disease, Alzheimer's type dementia. So the point is, it's important to understand what part of memory is being affected here, so that when we go to humans, we can recruit the perfect individuals that would allow us to see a signal. And here, in the third sentence, I emphasize this. Short-term memory is impacted in the early stages of dementia, but processing spatial cues to form memories becomes impacted in the later stages. So this also weighs into when in the process of memory impairment do we intervene. So our solution, we actually have a nice way by measuring the process of memory formation and storage using what's called electrophysiology. So this is, I guess, the best example I can give you is Neuralink, Musk's company, where they implant a chip in the brain and it helps you control a lot of things. It's the same idea, except instead of implanting the chip, we're implanting electrodes and we're sending signals through these electrodes and we're picking up the signal in different parts of the brain. So what did we do? We implanted the 2 electrodes, one in the entorhinal cortex and one in the hippocampus. So what's the EC? The EC is all your thoughts and memories sort of go to the EC. It gets packaged nicely into a nice FedEx package that then gets sent on a nice FedEx truck called neuronal axon all the way to the hippocampus. And then the hippocampus stores it. So memory, you can think of it, the process of memory formation consists of 3 steps. One is delivering the package. Forming and making this nice package and delivering it. It has to be nicely packaged and delivered perfectly. without being damaged. So that's the first part, the packaging and delivery. The second part is storing it. The hippocampus receives it and says, okay, let's store this piece of information. And then the third part is retrieval. You know, this whole thing we all do of scratching our heads and trying to pull out a memory, that's retrieval. For our study, we just looked at the packaging part and the storage. We haven't really looked at long-term retrieval yet, but we will be doing that at some point. So again, we implanted the same rats with electrodes in the entorhinal cortex and the hippocampus. We sent a small electrical stimulus mimicking a memory through the entorhinal cortex, and we basically looked at how this signal transferred to the hippocampus, and then whether it was stored or not. And we look at storage by sending the same signal an hour later to see and we look at the voltage potential. So here are the results. Let me explain what the y-axis means. Actual over predicted evoked response ratio. So actual is basically the signal we send, the memory we send. And let's say we send 10 millivolts of memory. We predict in 1 hour, if memory storage was 100%, we would expect 10 millivolts. We should expect a signal of 10 millivolts to be seen in the hippocampus. So that's why we call it predicted. So what we did was we measured the actual response, which actually mimics the packaging. And I just have to say there's no changes between the groups in the packaging of the signal. So the high-fat diet doesn't affect the packaging. But what the high-fat diet does is it affects the storage part. So what we're saying is that the hippocampus receives the memory, okay, everything is fine. But what happens is when you have the rats on the high-fat diet, they can't store the memory well, as you can see in the orange scatter plot here compared to the low-fat group. When the high-fat diet contained the WPPC, in some rats, this was partially reversed, but in others, it didn't. So that's why we're saying there's some improvement in connectivity and memory storage by the WPPC, but that's not all. And that's why we say it's partially reversed, but I think there's more to the story than the WPPC affecting the storage part. So future direction. So just to reiterate here, what we're seeing is that the high-fat diet, and we actually didn't know this in the past, it doesn't affect the packaging of a memory, it affects the storage of a memory. And the WPPC partially reverses that, so by improving connectivity. So if you're sort of improving this ratio of actual to predicted evoked response, it means more neurons are talking to each other, more neurons are shaking hands, and that's a good deal. But in some animals, not all animals. And we need to understand why. So we think there's other factors at play. So future direction, I think more work needs to understand what components of the WPPC are working, but I agree with Ben, Nurit, and Marie, and everyone when they say the whole is better than the sum of the whole is better than the individual components. We're just interested in what components are working from a pure scientific perspective. But hey, I mean, the whole package is working, so why do you want to ruin something that works already? And then the second aspect is we're looking now to understand, and I think we'll have this data in the next 2 weeks before we publish this paper. If the WPPC is modifying brain inflammation, that's another mechanism through which memory could be improved, or structure. So it's sort of restoring structural deficits that the high-fat diet is known to induce. So atrophy, for example. So we'll have an answer in 2 weeks, but I don't have the results yet. Also, the question of memory retrieval needs to be addressed. Remember, there are 3 parts to memory: packaging, storage, and then pulling it out, especially a few days later. We can do that. We just didn't think of doing it at the time. We're just super focused on the packaging and the storage. But I think, in the next experiments we're doing, we're going to be looking at retrieval. So if I send a memory on Monday, do I retrieve it on Wednesday? And this is synonymous to what's called the long-term delay recall in humans. It's a test for cognitive that they do in cognitive testing. And that's also impaired more so in Alzheimer's type dementia. We're also, and this highlights what I was saying earlier, ongoing work, we're looking at brain phospholipid integrity because that feeds into the structural hypothesis. Again, the idea is that WPPC is high in phospholipids and so is the brain. And that's why we think the high-fat diet is, the WPPC is modifying brain structure at the phospholipid level or even the myelin level. And we'll have the answer in 2 weeks. But how does this matter to humans? So we just finished, published a human study where we did, the left side, the left column is Christmas lights. It's different types of phospholipids that we see in human plasma by using lipidomics. I don't want to be dazzled by it, but just to say that a lot of these phospholipids can come from different food sources. One of them that stood out is PC17017. This comes from milk, actually. And we have data showing that it's highly concentrated in WPPC. And in our human cohort, where we looked at people who have both Alzheimer's disease and vascular dementia or cerebrovascular disease, we find a strong positive association between blood levels of this phospholipid, which would potentially come from WPPC or milk, but to lower levels, of course, in milk, and memory. So higher levels were predictive of greater memory. And that's exciting. That sort of fits into the phospholipid hypothesis we're working on in the rats. We saw cognitive improvements in the rats with the WPPC, and the question is whether it's occurring by altering the plasma phospholipid structural profile, especially some of these phospholipids that are found in milk. And then, of course, the fatty acids in them get cleaved in the plasma. The brain takes them and reconstitutes them in the brain. So we're exploring these questions in our rats to see if this is one of the mechanisms. Future directions, again, WPPC may enrich some of these phospholipids in serum plasma that relate to better cognitive health. And we're really doing some work to validate this hypothesis. So it goes back to what all the prior speakers were saying. It's the structure. The structure gets digested, and it leads to more structural phospholipid uniqueness and diversity in the blood. And then the blood sort of supplies it eventually to the brain indirectly, of course. So conclusions. WPPC, a byproduct of cheese and whey processing, is highly enriched in phospholipids that could yield fatty acids the brain can utilize. Again, the brain doesn't take up phospholipids, doesn't trust anyone but itself. It takes the fatty acids and makes the phospholipids itself. But we do need consistency in the processing methods, especially if we go clinical. We can't give patients, 1 week, WPPC that has 4% phospholipids, and then another week where they have 8%. So we got to be consistent. Preclinical data suggests that the WPPC at either doses supports brain health by preventing cognitive decline caused by a high-fat diet. Mechanisms involve partial improvements in memory storage. Other unknown mechanisms are being investigated. And we're also doing studies now to see if we can reverse the cognitive decline. So what I showed you so far is prevention. The next question we're addressing is if we get animals who are cognitively impaired, can we reverse the problem or stabilize it? Again, our goal is to prevent people from going from MCI to this vascular dementia or outright dementia stage. With that, I want to thank people in the lab who contributed to these studies. Daniela Baril, who sort of pulled me into this, which is amazing. I'm a believer now, I'm a supporter. And then my funding, and I appreciate your time and ears. Thank you very much.
That is so spectacular. All right, so the formal presentation portion of this spectacular event is now concluded. We will take a 5-minute break where I will be getting some WPPC to assist me in moderating the discussion section. And I can't wait to ask questions. We're just getting everybody back into the Zoom visible stage. We have more questions than we will be able to handle in the hour, but we will be covering most of the questions over the next hour. Many of the questions are speaker specific. We will ask those, but we will also ask some of the questions that turn out to be more general and are appropriate for everyone. I'm going to ask a couple of questions quickly of the different speakers that are very pointed. So, Marie-Caroline?
Yes.
Andreas Moeller asks, have you looked into how native milk fat globule structures of different size impact gastric emptying rate, satiety signals, and diet-induced thermogenesis? Do you think there will be a difference between native structures and disrupted structures?
We did not dig into that point. However, I can probably provide some elements of answers from either our own work or international colleague works. The milk fat structure and or the presence of the milk fat globule membrane, we showed in adults that this can modulate the lipid beta oxidation after a dietary intervention. So there may be some aspects that can finally modify the final postprandial lipid beta oxidation metabolization. Regarding the structure of the droplets, there are preclinical studies that are more related to the infant nutrition field, showing that small artificial, let's say, droplets do not lead to the same fat accumulation after a high-fat diet or to the same indices of mitochondria activity compared to milk fats, fat droplets that would more mimic the structure of the milk fat globules. With mechanisms that still have to be digged into, but there are some preclinical work on that and some first human studies as well. In humans, there have been 1 or 2 clinical trials on postprandial metabolism after native versus homogenized milk, but it was not very conclusive. However, for long-term consumption, I think the question is still open. Because of all these mechanisms that were discussed during the meeting, and I think also about what you presented, Nurit, about this interaction between bacteria, and there are bacteria also in the gut, so we are always discovering new mechanisms, so I think it's a hot topic.
Merci, Marie-Caroline. So I have a question for Nurit, and the question is a very pointed one. Do you think your tannin results in goats can be transferred to humans? Would you think you'd see the same effect in humans?
Wow, great question. I have to say that we, in this in vitro studies, that we took either standards that we could purchase that mimic some of the components in this tannin group and apply them on cells. We got similar results in terms of the lipogenic capacity, which was improved. Actually, we also saw some proteins, improvement in protein synthesis capacity, and also the composition of proteins. So it's not only about the anti-stress response of the cells when you apply antioxidants. So part of the tannins, as I explained, are polyphenols. Polyphenols are strong antioxidants, and therefore they can change many of the processes that occur in the mammary gland. But I guess this, and the question is specifically on omega-3 fatty acids, and I do not want to promise anything, but it definitely showed similar effects when we used in vitro models, but we still have to show it in vivo in a monogastric, at least monogastric model. So not maybe in humans, but in monogastric animals.
Okay, thank you. And a question for Ben, actually multiple questions, but one, do you think phospholipid digestive products also play a role? And could you elaborate a little bit on the magic 7?
Yeah, sure. So phospholipid digestion products will definitely play a role, but it's going to be in proportion to the total amount that's there. So as I sort of highlighted, the fraction of the total amount of lipid that is phospholipid is, not homeopathically low, but very low, because it's intended just to occupy the interface. It's not meant to be the bulk component of the fat droplet. So the bulk of the fat droplet is the triglycerides, and so the global structural attributes of the fat droplet during digestion are dominated by the triglyceride behavior. If we were to look specifically at the interface and structures that form at the interface, then we know there's a trilayer there at the start. Who knows what happens locally, really, during digestion? I mean, you'll lose some of the phospholipid because it'll be digested to lysophospholipid and fatty acid. So it probably just gets depleted of phospholipid over time, and it doesn't, doesn't change to another self-assembled structure, but we don't know yet. Second question, the magic 7. So, so that was an interesting exercise, and to reduce a year's worth of work to one number. So the magic 7 are 7 of the more common fatty acid species overall that make up the overall fatty acid composition. But when they're assembled into homotriglycerides, so all the same chain length in the one triglyceride, but when they're digested, of course, then you get a mixing of all of those monoglycerides and fatty acids, and potentially some phospholipid, of course, to end up with a composition that structurally behaves the same as human breast milk during digestion. So it's if we want to start adopting the mantra, at least that structure plays some role in transport of nutrients and transport of drug, in my case, because I'm from the pharmacy school, we know that is true. So it's getting to the finer detail and nuance of that's that's the task, and deploying some of these substitutes in a therapeutic or a nutritional challenge is going to be the next real step, I guess, in our work.
Fantastic. Thank you. All right. Amir, we have lots of people asking if they can get WBCC for themselves. But one of the questions has to do with, you mentioned that the brain doesn't let the body make its phospholipids, that it makes the decision. And you made the point about fatty acids. Is it also possible that the phospholipid portions that are left after enzymatic hydrolysis also could affect the brain composition by virtue of uptake and pushing those pathways?
Absolutely, and I think that's what's probably happening. There's a lot of I think we all need to get together and track this whole process with tracers from the beginning. Like, emulsify with the phospholipid, the phospholipids we characterize, and then see where things end up. But 90% of the brain lipid pool comes from the free fatty acid pool. So if you have phospholipids in blood, they need to be hydrolyzed to provide the free fatty acids. There is evidence showing that in people with mild cognitive impairment, this process of hydrolysis is impaired. And it could be because it's enzymatic, or you just don't have the substrate, the right substrate that sort of keeps keeps the enzyme going. And probably the WPPC, our working hypothesis is that it's just doing that, providing the right substrates so that the brain takes the fatty acids it needs and reassembles them. But again, the brain is a thank you, but no thank you kind of organ. It's like, okay, I'm just going to take the building blocks and build it myself. I'll waste 2 ATP molecules, which are expensive energetically, to do it myself, and that's how the brain works. So yeah, I think it's the substrate question and releasing these specific fatty acids. Some are unique to milk, by the way. So like the 17-0-DIPC, in a lot of clinical trials, 17-0 is the marker of dairy intake, actually, right?
Right.
So, and when you have WPPC, we found that there's a 10,000-fold enrichment in this specific phospholipid, which is why we're excited and interested by it. So.
Yeah, that's a nice signal to noise. 10,000.
Yeah.
Okay, so I will ask a question to everyone that they can answer in turn. So one of the things that emerges from this science is that the ability to make small native milk fat globules seems to be really important. And also data suggests that a metabolically compromised mother can't make as many small globules. So would you imagine it possible that for human mothers during lactation, they could get a measurement of the quality of the milk, of their milk, from this criterion? And if it was compromised, what could they do? I know it's wildly speculative, but I really think this is where mothers need to know the quality of their milk. And if it's not as good as it could be, the implications are both to their infant, its neurological development, metabolic development, and to the health of the mammary gland and protection from mastitis. So starting in turn with the order, can anyone come up with ways to measure this dimension of milk quality? And what would you suggest to mothers if they knew they wanted to improve it? So Marie-Caroline?
Thank you, Bruce, for this challenging question. I was beginning to elaborate here inside my brain that is obviously fed with milk lipids every day. On your question, and based on what you presented, Nurit, really brilliant. I am a fan of all this mastitis interaction with bacteria. I think you really nicely demonstrated. So it seems like in these animal models that there can be a modification of the structure of the milk fat globules according to the, let's say, contamination status or health status initially. On the other hand, the smallest the globules, maybe within a certain limit, the smallest the globules for a certain fat contents, the more the milk fat globule membrane material, which on a different population, not in a health problem topic, maybe it's not so bad to have a lot of milk fat globule membrane material because we saw some metabolic benefits, and Amir very nicely saw also impacts potential link with brain health. So maybe this the point is that is really monitoring the size, something that should be corrected in some way. Maybe it depends on the health context of the mom. Maybe there is not a single criteria. What do you think, Nurit?
Yeah, I think you're definitely right. I don't think that one size fits all. I think that is really depending on the health, maybe metabolic status and other health conditions that the mother can have, and also for the neonate. In terms of what regulates size, I can think of several aspects that we found throughout the years. Unfortunately, most of them are related to stress. So I don't think that stressing lactating mother should be a strategy that we want to implement. But it seems like that the mammary epithelial cells react to stress by reducing the size of the milk fat globules. At least in our in vitro studies, this is something that returns over and over again in different kinds of stressors that we use. But maybe also the change in the milk fat globule size is something of significance. So maybe not having the same structure all the time, but changing the size can signal something, again, either to the mammary gland, the environment within the mammary gland, and the end consumer, which is the child, the neonate, or even an adult consuming milk fat globules. I can also say that maybe if we think about something like nutritional strategy to maybe change the milk fat globule size, so we try that, but we don't have like absolute certain results. Maybe it's components like choline, for example, can change the milk fat globule size. It was shown in the liver, for example, it enhanced secretion of lipids from the liver. So maybe it has something to do with more membrane material being produced, and therefore you can reduce the size of lipid droplets and have more envelope material to stabilize it. And certainly the mitochondria here, which takes me back to the stress and your question on, on the goats and the polyphenols and the tannins. The mitochondria plays a very central role, as again, from our studies, this is what we see, both navigating between the stress and the output of the mammary gland, which we have to understand. It's not like adipose, for example, that you can change lipogenesis, but eventually the lipid droplets will stay inside. The reason for producing fat, so to speak, of the adipose tissue is to accumulate the fat. And here we're talking about a tissue that the aim is to deliver fat to the infant. So again, the mitochondria plays a central role here. And all of these aspects should be studied by more people with more money and more resources, for sure.
Yeah. And Ben, can you also talk about the implications therapeutically of being able to deliver lipid-soluble therapeutics for a broader audience?
So the reason, I guess the reason I'm sitting here, Bruce, as you know, is because we were already looking at drug. So drug needs to be dissolved to be absorbed from the gastrointestinal tract, and lipid formulations in the pharmaceutical context are one of the tools that we use in order to get drug into a dissolved state so it's available for absorption. Of course, nature's been doing that, since the year dot, in the form of using lipids, at least in some form, as a vehicle for keeping poorly soluble vitamins and other nutrients in solution, so they also are available to be absorbed from a solution state. So it makes sense that we should circle back and look a lot closer at using milk and similar systems, with the deliberate intent of it being a drug delivery system and a nutritional delivery system, but using some of the more contemporary tools that we now have to understand the interactions of drug and poorly soluble compounds in general with these, with milk fat droplets and fat droplets from other types of liquids. So that's another large part of our research that I didn't have the time to really go into today, but it's I mean, one of my students from Monash is here in Copenhagen doing a oral bioavailability study on a polysoluble drug delivered in a range of different milk and milk-like systems in piglets, looking at it from an infant delivery perspective. She's got some amazing results hot off the HPLC today, actually. So, it's a big area, and I think there's really exciting things to come.
Oh, thanks. Yeah. Now, Amir, in answering that small globule question, can you also respond to multiple questions from the participants? If you were going to design not what's available, WPPC, but completely revolutionize milk processing to maximize that which you think would be therapeutically effective, what would you imagine? Even including, what would you feed mothers?
A very loaded question, but I'll start with so one of the things, and I didn't mention this in my talk, it was focused on the phospholipids, but there are, with Professor Daniela Beril and myself, we published a paper on the detailed composition of the WPPC. There are other things there. There's protein, of course, but the diets were matched in protein composition. So I didn't mention, but the low fat and high fat did have the whey protein components. But there's also oligosaccharides, and we're very excited in exploring this hypothesis because oligosaccharides contain some precursors that can actually enter the brain without any strings attached and accumulate there in specific axons and so forth. So we are exploring that, and I saw that was one of the questions. And then another one was on the cognitive impairment secondary to metabolic dysregulation. Yeah, we think there's truth to that, and we're also exploring whether the WPPC is modifying metabolic parameters, leaning on Marie Carolin's work. And then in terms of the reconstitution, I think it goes back to the question of what's really driving all this, and we know what the WPPC contains. So we're trying to dissect to figure out what's driving it. I'm in the line of thought that the whole is better than individual components, but I also think it's important to understand what's the most significant contributor to all these changes we're seeing, whether it's metabolic or cognitive. And then if we figure that out, then we can start to modify potentially infant formula to reconstitute it with these fractions or stop parts of the fractions to sort of compensate for differences. But one thing I want to sort of throw back at all of you is using milk fat globular size to diagnose maternal health status, right? And this the same extent as Nurit was mentioning that the globules tend to increase with metabolic impairments. We can use that information to look at the diversity of size and composition maybe to diagnose things that go undiagnosed postpartum.
Yeah, I agree 100%. If we could provide anything from the science that you all have done to improve the human condition, it's being able to monitor the quality of the health of mothers, to be able to know, is milk being successfully assembled, and therefore, is it the best for that infant? Several people have asked about that. What's the difference when you look at an infant gut and its capabilities and an adult gut and its capabilities? Has anyone seen significant differences, or can we say that we're all babies at heart or at the gut? Marie Caroline, they specifically asked about your studies. Could you imagine a difference?
Even though I did not study the fate of the milk lipids in the infant gut, I think the
the key point in infant gut is the image, of course, obviously the initial immaturity first regarding digestive, enzymes, etc., which is well described, and the benefits of the lipase associated with human milk, by the way. But also there are more and more data about the immune development of the infant gut, the development of the gut barrier, the maturation of the gut microbiota. So in fact, there are several aspects in the differential potential sensitivity of the infant gut to potentially different lipid structures that are related to this evolving maturity, which is different, I believe, from the adults, even though an adult gut is also constantly in a renewal process, because every other week we have an entirely brand new intestinal mucosa.
Yeah, I think the difference between infants and adults is so complicated. We're going to have to, as many of you are, you said, encourage research into this direction and pursue more research. I'd love to see endogenously isotopically enriched C13 in all milk components.
Yeah, of course, obviously.
And one of the great outcomes from cellular models of milk synthesis could be the elaboration of C13 enriched study materials for clinical trials. The ability now of mass spectrometry to be both sensitive and accurate to follow metabolic pathways, fatty acids, etc. It just will change this field, I think, dramatically. To that point, if you were going to speculate, all of you, to the technology that would be most disruptive of this field, that we should be encouraging the scientific community to pursue, is it, to Ben's point, of real-time X-ray? Could we imagine someone coming up with real-time NMR? Or, I mean, what would it be? How do we get Google to direct AlphaFold towards lipids? What would change the scientific community's perspective on lipids to be able to say, oh my gosh, we've missed where structure is really important. It's in the lipids. So what techniques do you think we should be telling NIH, start putting money in? INRA, all of them. Ben.
Yeah, so not everyone's going to have a synchrotron in their backyard, but we have been working on alternative spectroscopic techniques that are sensitive to structure. So low frequency Raman is one technique that's really starting to emerge as quite an interesting opportunity to look at structure in systems, because although it's spectroscopic, it's related to sort of whole crystal vibrations, not bond vibrations like most spectroscopy techniques. So, and the instrumentation for that is really only now becoming sort of a commercially viable thing. So we've been starting to look at lipid self-assembly using that technique, and I think there's a huge amount of exploration and investigation that should go on there, and there's nothing to stop you having a handheld low frequency Raman instrument once you know what to look for.
Wow.
So if
every NICU should have one.
Sorry?
Every NICU should have one.
Every NICU could have one. So it will get to a stage where it's just like a handheld Raman, normal mid-frequency Raman unit that they use for testing counterfeit drugs, for example. The low frequency version of that is only a matter of time. So it's what you actually measure, and why it's important is that we don't know that yet. But I think there's an opportunity to certainly find out. So
Cool. Nurit, what would you like?
Yeah, I will just pick up from where Ben ended his answer. So remember, I'm more lactation side of looking at the lactation side of things, the physiology of the production of milk, and we're actually using Raman. So we had quite a big project on using Raman to explore differences in lipid composition at different sizes. So very straightforward, easy to manage, very cost-effective method to study a variety of lipids all together in one analysis. And we're looking at actually using nanoparticles, gold nanoparticles, to enhance the Raman signal and study how the membrane of the cells changes according to different, again, stressors or metabolic conditions of the lactating mother and the mammary gland, and how it changed the kinetics of lipid secretion. And part of that is also the size of the milk fat globules that are being secreted. So I think it's nanoparticles enhanced spectroscopy. Using Raman spectroscopy is the way. This is the machine I want in my lab, the equipment I want in my lab. That's for sure.
Just send someone up to Copenhagen.
Marie Caroline.
Well, I was thinking there are several developments in the past 10 years in vivo imaging, even, you know, for really clinical diagnosis and applications, but that would be really, yeah, great developments to be able at some point to visualize and track in the real life moment babies the fate of all this through adaptation of these techniques with specific probes maybe, or like, yeah, so this would be really the ultimate way. And also there have been several developments on ingested capsules, monitoring all types of signals within the digestive tract, and probably there can be improvement, enhancement, and use in this specific topic.
Yeah.
To enhance our understanding of what happens there in our gut.
Okay, Amir, what would you want?
I think definitely different types of mass spectrometry, because I feel they yield complementary information. You know, using the untargeted discovery type platforms followed by the targeted, where you know what you're looking for. I think all these tools could help us address, I mean, there's still fundamental questions in my mind that haven't been addressed. What's our baseline? What does, you know, normal look like, and where can we go from there? But we need to know it's the size and if there's any two words I would echo, it's the size, the composition, and then complemented by tracer studies and maybe Raman spectroscopy to understand you know, where things go from there. So I feel we're at the early stages, but hopefully one day we'll collectively resolve all of these unknowns. It's a black box in my head, I feel. But in terms of equipment, definitely mass spectrometry, because that helps us identify structures and all the heterogeneous compositions in milk. So.
Wow. Okay, now I have a question for all of you that has to do with milk as a product. So if you look at the history of milk and its processing, it's been processed almost exclusively to improve stability. All right, so you concentrate the caseins in order to make a drier product. You acidify with bacteria to make a acidic and shelf-stable product. We homogenize milk to improve the long-term physical stability. So it's basically a stability improving enterprise. But now it's clear that virtually all of those compromise the quality of the lipid structures. And so could you imagine if you're making a suggestion to the dairy industry worldwide, what kind of products would one imagine that if you were targeting, let's say, an aging population worldwide, how might you process milk so that the biological properties of the lipid components, both the composition and structure, would be at the very worst as good as the native milk, but even if you could go further to make them even more concentrated and biologically targeted to that population. So we're not going to tell the dairy industry how to do what they're doing now better. We're going to suggest, here's how you could do something completely different. And the value proposition would not be it's more stable milk, it's more therapeutic milk. So Marie, Caroline, can you speculate? What would you tell the dairy industry is the best thing they could do?
I think the lead can be towards inspired by a mix between all the information we had, part of information we had today, notably about the bioactivity of some components of the milk fat globules, their size and their potential native structure, and a combination with all the emerging increasing literature about the benefits of fermented dairy. And so for specific populations like the elderly, I think we also have to think that dairy is not only, there are the benefits of milk and its consistency and its components, but there is also the matrix and very often fermented matrix, and there are more and more information about how this can be impactful, notably by regulating the microbiota. And contribute also to the digestion and bioavailability of nutrients. So I think this is probably a combination of way to go to improve further the approach to nutrition and not only preservation.
Thank you. Nurit.
Yeah. I will refer to two studies that inspired me during my career. I think when I first started my postdoc in California, I read a paper by a French group about membrane separation of milk fat globules that maintain the native structure of milk fat. Of course, Marie-Caroline is one of the authors on that paper, if not the leading author, can't remember. But anyway, I think that definitely the industry needs to start paying attention to the macrostructure of the milk components and not disintegrate them as a first part of the process. Almost all of milk dairy products will start with disintegrating the lipids. I also was inspired by a paper that talked about the postbiotics, mostly on carbohydrates in milk, but how the bacteria basically metabolize it and what is the outcome and how it's affect the epithelial layer of the gut of the consumer. And we took this approach and used it to study the interaction of guts of microbes and fat. But eventually what we saw is that this fermentation process can result with an anti-inflammatory substance. The postbiotics of the bacteria can exert some anti-inflammatory effect on epithelial layers. And I think that these are the things that I would like to see from coming from the industry. So guide this fermentation substrates into something that is well, stable and safe, but also have bioactivity and significant bioactivity. And inflammation is one of the biggest problems, right, around the world in elderly, also in children, but also in elderly people. And I think this, at least some of it, can be alleviated by using fermentation tools on the right substrates.
Oh, absolutely. And the more people interrogate our microbiome ecology, it's clear we're all living in a post-antibiotic microbial world. Internally, we're going to have to find ways to deliver appropriate bacteria. And yeah, dairy should own that space for sure. All right, Ben, from a real therapeutic pharmacological perspective, what would be the products that would change drug delivery?
I spoke to a dairy company and said, you need to reinvent yourself as a pharmaceutical company, because because I did think that the opportunity that was there is really there for the taking. It's close to Bruce's heart, the idea of therapeutics for premature infants, and coming to grips with, so premature infants are often treated with drugs intravenously only because we don't really know enough about how to deliver them orally. But nature delivers poorly soluble nutrients orally perfectly through milk. So I think that if I was to pick one area where I would like to see development, and it's not necessarily going to be a huge commercial money maker, but it's certainly a market, and is arguably the most important one, is in designer milk products deliberately designed for the delivery of therapeutics to infants. And there's certainly an opportunity there, and there's certainly an unmet need, and that's really where we're going. So, but in the bulk market, I would say pay more attention to what happens once it's in the gob rather than in the bottle. So gob's an Australian word for mouth. So the consequences of processing on digestion are really just starting to be understood, and the more we learn about that, I think the more we're going to retrospectively be suggesting that processing is done this way rather than that way, or perhaps a slightly different focus on the presentation of products would make all the difference.
Right. Okay, Amir, take us home.
So I'll sort of, in the cognitive field, the dementia field, there's always the question of, we always talk about cognitive impairment, but there's the big question of what's normal cognitive impairment. There was a paper recently showing that, most people with MCI, mild cognitive impairment, go undiagnosed. And when I saw it, I'm like, of course, that makes sense, because we still don't know what's normal. If you know what's normal, then you can start to address what's abnormal and get it diagnosed and maybe treated. So it's the same issue I feel with milk. I feel in a year from now, I'll be better endowed to answer this question, because I know we're doing some of this work and other labs are doing it. But it goes back to the heart of what I was saying earlier. What's our baseline? What does normal look like, at least in terms of the chemistry? And then comes the question of how can we modify the chemistry through the tannins or other approaches to optimize the benefits, metabolic or brain. And that's where I think I am, at least with answering this question. I feel I'll have a better, more informed answer a year from now, maybe 2. But for now, I think the focus should be similar to the whole dementia field. What is normal? It's okay to have decline cognitively with age, but when is it not okay? Same things with milk, size, composition. certain sizes, certain contents may be okay, but when is it not okay? And that and these are the questions we need to address and guide from there. That's my 2 cents worth.
Yeah, well, I think it's clear that when you have mild cognitive impairment, you're made the moderator. So sorry. Yeah, I think one of the things that we should be looking for is for greater therapeutic impact, recognizing that the gut is a biogeography that we're only beginning to realize is important, literally from, as Ben says, it's gob to poop. Then we should be thinking about products that you can take all day. Every time there's a consuming opportunity, if milk products are going to be enhancing the biological activity, therapeutic efficacy, then once a day as a large volume isn't the secret. It has to be thought about is how would you manage your day to make sure that morning, noon, and night, the products are appropriate for that. So I think there really is a large opportunity for the most important biological beverage to really look at new opportunities to be therapeutically effective. So we only have a couple of minutes left. And so now, if there's one thing you want to leave all these young minds, so we have young people who are in the audience thinking, what should their career trajectory include? What would you tell them? They should continue to monitor the literature, think about incorporating into their research capabilities that they might not have now, but they could in future. What would your recommendation be to them? Okay, Marie-Caroline.
And even if, yeah, our current world is very sometimes speedy and on the tracks, keep open to sometimes unexpected interaction with colleagues having a very different expertise. Sometimes, I don't know, physical, immunological, microbiological experts that you would just come across and stay open to enriching, to unexpected approaches by crossbreeding the expertises, I would say. And of course, keep aware of the literature, yes. This is a basic that must remain.
Okay, well, and of course, you are all successful. You're all inspiring to these young people. And now we've got to, Nurit, what would you tell them?
First of all, I think they should know that the lipid arena is not crowded. So there are a lot of opportunities to really contribute scientifically and in terms of application to the world of lipids and specifically dairy lipids. And if I need to, pinpoint the thing that we don't know enough about, not in terms of functionality, not in terms of structure, and not in terms of the ability to analyze it, unless you have a very fancy equipment, these are the glycolipids, because these molecules seems to be very important in all kind of aspects, microbiome-wise, brain development-wise, etc. And there are not enough labs that can really analyze them on a regular basis. And I think this is some expertise that you can really leverage from.
Whoa, thank you. Ben.
And drop me an email when you succeed with it.
Yeah, I will. Ben.
Yeah, so I would say that, yes, it's good to do, you'll have your core interest in the area that you're trained in, but always be on the lookout for the analogy of different fields and different techniques might be used somewhere else. Bring it in, just throw it at a problem and see what sticks, because often you learn nothing, but you only need to learn something 1 in 10 times that really can open up a whole new area of understanding. So don't be afraid to try techniques from, or at least collaborate with people that are experts in techniques that you know nothing about.
Yeah. Oh, I echo that. Absolutely. Okay, Amir. Young
So,
minds.
echoing all of the above, the only thing I would emphasize is, looking at me, I'm sort of a brain guy. I work on the brain. Milk was the last thing on my radar. But it's through interdisciplinary research, Bruce daily telling me about milk, and Daniela Baril from my department, working with her to put the grant in. That sort of lured me into the milk field. And I'm not a milk guy, I'll admit that from the beginning. But it sort of lured me into it, and I'm really excited by it because, as Marie mentioned, and Bruce also mentioned, there's new opportunities there's a lot we don't know, and there's huge opportunities that need to be explored and addressed. So don't be afraid of interdisciplinary collaboration, if it's the brain guy next door or the heart person working together. I think this is where science yields the most impact. And measure, let me know when you measure glycolipids also, please.
And keep your favorite research question always in mind throughout your journey.
Yes. Well, this has been the highlight of my year. This is fantastic. I'm so excited to have been here. And in spite of mild cognitive impairment and moderate. Okay, I give it back to Aleki to wrap up.
Thank you. That was incredible. So much collective genius amongst all of you. I am inspired. I am motivated. I hope everyone else on this call is too. I'm going to quickly take 1 minute and wrap up. This was a fantastic webinar workshop with so much information. If there is any part you want to revisit, that is possible. We did record this session. It will be available on the IMGC website in May. As someone who attended the webinar and registered, you will get an email when that recording is available. So keep an eye on your inbox. We will let you know. Next slide. Really quick, just a massive shout out to our IMGC steering committee, as well as our scientific advisory committee. And then, of course, a big, big thank you to our sponsors. Without their support, this would not have been possible. So Arla, By Heart, the National Dairy Council, and then, of course, the California Dairy Research Foundation. I apologize, I mispronounced that during the intro. But thank you for your generous support to make this event happen. And then last but not least, if you are inspired like I am to get even more involved with IMGC after this workshop, we have a few ways for you to do so. Please scan the QR code to take the 5-minute exit survey if you have not yet been able to do that. We really want to hear your candid thoughts on what went well, where can we improve so that next year you will join us again for this roll up your sleeve workshop. Secondly, the top QR code right here will allow you to subscribe to our bi-monthly translational newsletter to have us pop into your inbox with all the latest translational information about the new discoveries in milk science. If you want to stay connected on LinkedIn, we do have a private LinkedIn group that please request to join. We will accept you and we can continue the conversation there. We do offer working groups on occasion where you can join us. If you are part of the Splash newsletter, that is a fantastic way through which you can be notified when workshop opportunities and working group opportunities become available. So another reason to subscribe. And then last but not least, I hope to see you live in Davis in October of this year for the annual IMGC symposium. Keep an eye out on the IMGC website because abstract submissions for oral presentations those have already, that submission period has closed, but abstracts for poster presentations are opening up really, really soon. If you have research in the dairy space, please submit. We would love to see it. That's that. Thanks again for joining, and we hope to hear from you and see you very soon.